Hello and welcome to our Lakeside chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segurate Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And so this is a place that my ancestors have been since the beginning of time. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
Thank you, Corinna. Those are inspiring words, and we know that we have a long way to go, but we appreciate all of the knowledge that has been passed to us and is being passed to us from our Native communities. So I want to remind everyone here tonight that Rotary Nature Center Friends is an independent, all-volunteer 501c3 nonprofit, providing online and in-person programming for all the people of Oakland. We engage the community in real science and stewardship activities, bringing nature and people together in the heart of downtown Oakland to steward the nation's first wildlife refuge, the Lake Merritt Wildlife Refuge. The focus of our activities is community science and environmental justice. And we'll be sharing some of these activities with you tonight. And we would like to say, take notice today that as we begin the month of February, that there are many important things to observe. February is the shortest month. It's also Black History Month and a 100-year anniversary of the observation nationally of African American history. It's also the 270 contains the 217th birthday of Charles Darwin. Charles Darwin was born on February 12, 1809, on the same birth date as Abraham Lincoln. He changed our view of the world and our place in it forever. Insights provide guidance to scientific research today. He was the author of 2 important books, or actually many important books, but The Voyage of the Beagle and The Origin of Species. And he liked plankton. On the Voyage of the Beagle in 1832, Charles Darwin made and used a plankton. He was the second person whose making of a plankton net was recorded in Western literature, and he was amazed by and fascinated by the beauty of plankton. I'd like to read this little passage of this. He said that many of these creatures, so low in the scale of nature, are most exquisite in their forms and rich colors. It creates a feeling of wonder that so much beauty should be apparently created for such little purpose. And he went on to ponder that for many years before arriving at his theory of evolution by natural selection. So happy birthday, Charles Darwin. At the beginning of each of our programs, we like to highlight a few of Rotary Nature Center Friends' activities during the past month. And we would like to thank everybody in the community who came out to volunteer for Martin Luther King Day, both for us and for all the organizations at Lake Merritt and throughout Oakland. It was a wonderful day of tribute and service. We would like to thank our new California naturalists for hosting a Mornings at the Marsh at Lake Merritt. and teaching the public about native plants and introduce plants and how we might take care of this location so that we can all learn and enjoy the stewardship of a more natural shoreline around Lake Merritt. And we'd also like to thank our high school New Voices, our Rising Water Justice Fellows, who have been following water quality and plankton in their 25-26 fellowship. And we also, in this year, are offering for the second time our Urban Estuaries Naturalist certification course, and that's available for enrollment now on Eventbrite. You can get an Eventbrite ticket, and we will follow up with the details of completing enrollment and so forth. We do offer scholarships. We want to make this program available to everyone who is interested in learning about Lake Merritt and urban estuaries in general. With that, I would like to introduce you to tonight's guest speaker, Janai Southworth. Hi, Janai. And it's my great honor to introduce Janai. She is an amazing person, as you will soon learn if you haven't met her before. I asked her what I might say about her autobiography, and she said, well, I'm actually a careerless person. And so that's kind of interesting, but she has accomplished amazing things. She has a Bachelor of Arts in Fine Arts, and then she spent a great deal of time in the School of Life, taking care of her family and pursuing many interests related to that important work. But then as she proceeded, she took many community college courses related to conservation and science, and was poised to take on a master's degree program in conservation, but life intervened, and she then continued to volunteer, and she volunteers today in community colleges. She is the president of the San Francisco Microscopical Society, and they do many wonderful presentations online and all throughout the Bay Area and beyond. She is part of the Greater Farallones National Marine Sanctuaries Foundation. She's a naturalist. She's on the board of Rotary Nature Center Friends, and that's
good. That's good.
Okay, I could go on, and I don't want to take up all that time with all the wonderful things. But anyway, thank you so much for joining us tonight, Janai. And what are you going to show us tonight?
I'm ready. We're going to talk about plankton, all the microscopic organisms that are hiding in the water in plain sight all around us all the time. So even though we're going to be looking at the microscopic world that's around us, because I really enjoy using microscopes, it's a passion of mine. Plankton doesn't have to be microscopic. So I figured we would start this off with a really quick set of slides that talk about what plankton is. And so here we go. Plankton is all of these things. There are plankton that are animals and plankton that are plant-like. But to really be plankton, you need to be three things, right? There are these three main criteria you must meet in order to be considered plankton. So the first is very basically, you need to be alive. If you're alive, you could be, that's the first criteria you fit, yes. Okay, but the second one is in the water, right? So plankton is alive and in the water. And while I enjoy the water, I don't complete any of my life cycle there, so I am not considered plankton just based on these two criteria, even though I enjoy the water. Like, you're like, oh, if you're swimming, does that count? If you're floating in the water, no, because you don't want to stay there. You're not going to live there for a part of your life, even if you really enjoy it. And the other piece, maybe the most important piece that defines what plankton is plankton are drifters. So a plankton is a drifter. So if they're alive in the water and they're drifters, they're considered plankton. And then if they meet all of those criteria, scientists love to split things into groups. So we're going to split those organisms that meet those criteria into two big groups, right? The phytoplankton and the zooplankton. And the phytoplankton, right? The root of phytoplankton is phyto, like photosynthesis. They're the plant-like plankton, and zooplankton, and it's got that zoo root where you would consider animals to be. So phytoplankton, what are phytoplankton? They're aquatic drifters, they photosynthesize, and really they're the foundation of the ocean food chain. They are the producers. They eat the sunlight and they turn it into food for other things. And the organisms that do that, some of the most common ones are diatoms, single-celled algae. There are dinoflagellates, also single-celled algae. But the dinoflagellates have flagella for swimming around. And there's another one that doesn't get very much credit, but is really interestingly important in the oceans, especially are the cyanobacteria, or the blue-green algae. Very, very tiny. One of the most abundant organisms on Earth is called Prochlorococcus. It's a tiny cyanobacteria, drifts in the ocean, tends to live in the tropical, the warmer kind of middle waters, not near the poles. Makes, through photosynthesis, provides us with an abundance of oxygen. But then there's the zooplankton group, and scientists like to split that down even further. So we did phytoplankton. Now we're going to quickly talk about the zooplankton in the two big groups. They were in the quiz questions. We've got the holoplankton. They're plankton for their whole life, and the meroplankton that are only plankton for part of their life. So the holoplankton, I like to think their whole life, you can think of them as plankton. They're tiny drifters. They float, they drift, and they never, through any part of their life cycle, just drop out of the plankton. They're always there. And then there's the meroplankton, part of their life cycle. So things like, we have our most common interactions with organisms that may have started their life as meroplankton. Things like sand dollars and sea stars, organisms like crab and shrimp and lobster, clams and oysters and mussels. So with all of those groups of things, the other thing that's really interesting about plankton is you can zoom in very, very closely and look at things up close. And here we have a diatom. Here's another diatom. These are common to San Francisco Bay and to Lake Merritt. This is a dinoflagellate here. You can see the groove where the flagella would be. Here's another dinoflagellate. And then you can also see plankton from space. And while you look at individual organisms on a microscope, from space you would look at huge blooms and shifts in the color of the ocean that's caused by them reproducing in vast quantities. But what really concerns us typically about plankton is how they affect our environment. I mean, they'll be doing their own thing out in the ocean. It's no big deal. But here, this is the organism that caused the harmful algal bloom that impacted Lake Merritt and San Francisco Bay that Katie was talking about. It's the Heterosigma akashiwo. Here's a little video. There were so many in the water you could actually see them clouding like a little tiny dust storm in a bottle. But that's just one aspect. And that's, even though we often see the impacts of harmful algal blooms the most, we benefit from plankton and our interactions with them a lot more than we're harmed by them with the harmful algal blooms. They power the food chains in the ocean. They're the nurseries of many of the organisms that we either eat or appreciate, that feed people around the world. And the populations are shifting. And I don't have data. I'm not doing research in a scientific institution, but there is evidence that as the temperatures of the ocean are shifting, the timing of the organisms as they reproduce and they thrive and they bloom are shifting in the oceans, and that will have impacts over time. So here we go. I am going to stop that share for a minute. Now, for this next bit, let's see if I can do this. All right, my next trick, I am going to try and share from a secondary camera, which I have connected to my microscope. Let's see. And we have to rotate through my cameras, and eventually we'll get to the right one. So let's see. It should be this one. Let's see. Is it not? One more camera. There we go. It's going to be this one. Ha! Okay. So this is a little bit of, it's going to take me just a moment here. This is a slide that has dinoflagellates on it from Lake Merritt from today. I went out and I collected plankton. I'm going to play with the camera for just a moment so that it's optimized for you all. Turn that up a little, and we'll go back here. Let's see if I can get them in focus. So, of course, earlier today I had them, there we go. I had the opportunity to go and collect plankton at Lake Merritt, and I used a plankton net with a very tiny mesh. This here is, oh, it's a villager. We have a baby snail as a guest today in the plankton. I'll try and zoom in one more. So this is going to be a little bit of a live exploration to see who's here. Unfortunately, it's not pre-recorded, so you will have to bear with me as we focus in and out on some of these organisms. But one of the things that that I didn't say about plankton in that little flash intro, plankton isn't just one organism. It is a bunch of things that are alive in the water and drifters, as long as they meet those criteria. And that's why we have so many different, let's see if I can get this a little brighter for you all. There's a lot of dinoflagellates here. All these little moving beads and balls, those are all dinoflagellates from Lake Merritt. One of the things I didn't say though, which I was getting to, is plankton can be large. Plankton can be very large. In fact, they can be the size of giant jellyfish. So it's the alive in the water and the drifter. And while I don't have a facility here to look at the very largest plankton that there are, I do have a microscope. It's an Olympus that allows me to look at the tiniest organisms. And I mentioned to Katie that I was willing to stick around after this part of the presentation and answer some more questions and maybe do some more exploration, because microscopes and slides don't always cooperate live the same way slide decks do. So this organism here, I'm gonna pop to my other window so that I can, this one here, this is an organism called Noctiluca, and it's a sea sparkle. These tiny little balls that are zipping around, they're other dinoflagellates, probably a lingulodinium, but I'm not positive. And so this is a dinoflagellate, this tiny one, and this big one is also a dinoflagellate. They have a lot of different shapes and sizes. Let me drop that back off over here. What I'd like to do is see if I can find that little veliger, that little baby snail again. So I'm going to scroll around. We're using an objective that is magnifying things 100 times their actual size. If you were looking, there's that snail. If you were looking through the microscope, what you would be seeing would be 100 times larger than actual life. So this tiny little baby snail that's decided to zoom around here, let's see, right there, it's going to keep zooming in circles. Well, if it calms down, we can focus on it a little better. It has a rudimentary shell that's very thin. It's made of mostly calcium carbonate, and if it's slowed down, we could look at it with polarized light, because it does sparkle with the polarized light. But it also has a structure called a velum, which have little wiggling cilia, which are like hairs, that it uses to swim around. And while it's very young and in the plankton, it can move, but it can't swim faster than the current. So there's our little snail friend. We can see. I have a couple other slides here. I have one more from Lake Merritt up here. I'm gonna trade out. This one's very sloppy. It's quite a wet slide. Give me a moment. Here we go. If I can get that into focus. We get to start to see some of the different organisms here at Lake Merritt. So this is, we've got, they're moving so fast. It's a very lively sample, which is a good thing. It just means that focusing in on them might take, oh, there we go. That's a baby barnacle. Hold on. Let's see if I can get it back.
Yes? I'd like to encourage people to put questions in chat if you'd like to.
Great.
Yeah.
Yes. And then if you'd like to read them out, because when I'm using the microscope, I have a very hard time splitting my attention between the microscope and the words. So please jump right in with the questions. If you could read them out, or if Betsy wants to read them out.
Here's two great questions. Anthony asks about the flagella that causes, is it the flagella that causes their movement, or are there other means that propel them through the water?
So different plankton move in different ways. Dinoflagellates have flagella, and the flagella are what is propelling a dinoflagellate. There are any number of other organisms here today. Oh my goodness, sorry, I'm trying to move my light so I can see a little bit better. Unfortunately, it'll take me out of the light. There are organisms that have cilia, little wiggling hairs, like on the edge of the structure that the baby snail uses to swim around. There are organisms that have like a tractor tread of mucus. There's some of the diatoms that'll move that way in a very different style of motion. Jointed appendages that work like oars can be used to move around, because it's again, it's not just one thing. It's a lot of different types of organisms here. Let's see if I can.
There's a galaxy here.
Wants to know if Lake Merritt has a beneficial population of plankton.
A beneficial population of plankton. So beneficial to who? And this is, and I do this, and it's because they're existing, they're beneficial to themselves. Are they doing work for us by supporting a healthy ecosystem? Yes. But beneficial is a really broad term. I mean, they all have a niche in the ecosystem. So there's just so many here today. There's a copepod. Sorry, I'm distracted by the amazing plankton. And there's rotifers zipping around. So yes, there is a dynamic community of plankton at Lake Merritt, and there is so many on this slide. I can't focus on them because they're swimming around.
Alexis wants to know about, for the holoplankton, do they have sexes? How do they reproduce?
So nature doesn't keep it simple. Nature is complicated, and the reproductive strategies of organisms in the water is incredibly diverse and doesn't correlate neatly to what the standard gender-based strategies that people like to apply. It's not neat and tidy. There are diatoms that have multiple mating types, and they're the phytoplankton. There are some rotifers that have been have had one single gender for millions of years. So it's not straightforward always, and it's an incredibly diverse group of organisms. So if you're interested in reproductive strategies, pick one thing and look into that. But there's a plethora of organisms that live in the plankton. I'm just trying to get them to. They won't hold still because they're so alive.
So many of them. It's so cool.
Yeah, it's kind of busy. Any other questions while I'm here on the Lake Merritt plankton? I'm going to switch over in a moment.
That's all.
Copepods.
That's all I see for you right now.
All right, copepods. We have copepods. This is a juvenile copepod, Colonopleus. Here's another one. That is a polychaete worm larva. There's a bunch of the polychaete worm larvae. Let's see. If we have time afterwards, what I'd like to do is I'd like to have a slightly less active batch and try and get them to look at them really, really closely, because I think that you miss. Let me see if I can turn this down. Hold on. Managing the light on a microscope is a constant dynamic battle. There are those copepods. There we go. So this was collected at Lake Merritt by the sailboat house today around 1 o'clock. The tide was up because the walkway out onto those little docks was practically flat. So there was incoming water into Lake Merritt, enough to raise the water level. And I'd like to, I think this is it. Let's see. Switch to some. I'm trying to see. I had a slide that I made ahead with an organism on it, and I was just checking to see if I could find it really quickly before. No, I don't see it. But I do have bees. All right, so we're going to move to San Francisco Bay now, at least for a moment. So goodbye, Lake Merritt, temporarily. One of the things that people really enjoy about plankton, one of the secondary effects, things that plankton do, they can bioluminesce. And these organisms that are from the mouth, near the mouth of the bay. So I collected these probably around 2:30 from near the Golden Gate Bridge in San Francisco. There's a structure called the Torpedo Wharf that goes out onto the bay. And that's where these were collected with the same size net, the same mesh that I used at Lake Merritt. We saw one of them earlier in the Lake Merritt sample, but there were a lot more of them floating in the surface of my San Francisco Bay sample. The two bodies of water are connected. These here are sea sparkles. They're these giant dinoflagellates, and they're called sea sparkles because they bioluminesce. Let's see if I can. There's a diatom right next to that sea sparkle too. So this here, this is a diatom called datillum. It has cell walls of hydrated silica or opal. And this here next to it is that dinoflagellate, the sea sparkle. This I sampled from the surface of the sample jar because they tend to float. The Noctiluca do. Here, this is a great view. Noctiluca, which is what this is, the sea sparkle, they have these long structures coming off their single cell. It's a single-celled organism, and it's called a tentacle. It's not their flagella. Their flagella is tucked way up, very close in, and is hardly visible anymore. Let's see. We can maybe get a little bit closer here. Let's see if it'll, every once in a while we can see the movement of the flagella if I get the lighting just right. No. All right, we can try and go to bright field. Let's see. Phase. Sometimes with this lighting method, it can be easier to see certain structures. I can play with the light again. So one of the things about microscopy is that you really want to look at things in as many different ways as possible. Don't pick just one, because there are many, many things to discover if you look using different types of light and different techniques. So I'm still not able to see it, but you can see different structures now. This is a type of illumination called phase contrast. And it allows you to see certain structures highlighted in a different way. That's a big bubble up there. Let's see here. We'll try this one. See if we can find that flagella. It's easy to see the tentacle, but...
What's the function of the tentacle?
So, it has been hypothesized, I've never seen it in action, that the tentacle, as it waves and moves, will be dragging a thread of mucus through the water. And that thread of mucus through the water will catch things, and then it'll bring it back and suck it into itself. It doesn't really have a mouth. It just kind of like envelops it. I've never seen it happen, but that's one of the explanations for why they wave that tentacle around. I just really wanted to find that other organism because it was so very cool. I don't know if I have it anymore on this slide. I will try one last thing before we move on, because with there's just so much to see, so many different samples. Let's see, is this? There we go. I'm gonna try. I have, if it works, I have a hydrozoan. A hydrozoan is like a jellyfish. It's like a jellyfish. And it's very tiny. And I'm gonna see if I can get it to come out of the jar onto a slide. There we go. And I will be extra gentle so that we can see it without a cover slip. I'll have to be very careful with a microscope. So this is normally a no-no, but this animal has such a thick dimension. I just want to make sure that we don't. Here we go. We'll go out to dark field. There, let's get it in focus so we can see our little jelly. You can see it has tentacles around the outside. Oh, it's pulsing. And I'm gonna give it a little oblique illumination so we can start to see some of the other structures. There we go. So here we go. This is a hydrozoan. It's so big that it barely fits into the field of view. It has tentacles. Up here, these are the tentacles. This would be the top of the bell. I'm going to try and drive it around for a minute. So there's the top, what would be the top of the bell. And there are a little ring of tentacles here around where it's open when it pulses. It's just upside down right now. This is would be the stomach area up inside. And it's a little hydrozoan. So they will, part of a group of organisms, cnidarians. They have stinging cells. This one doesn't have stinging cells strong enough to sting people, but if you're food sized for it, you would be fair game. So something that has such a thin membrane, like that, maybe that baby snail or those tiny little copepods, it would be able to harpoon those very tiny organisms and capture them and eat them. So right now it's been eating a lot. It seems to have a lot of material in here because when I did the plankton toe, there were a lot of food-sized organisms for it to enjoy. So it's kind of special. There's
Hmm.
We don't often get them in the plankton, but I am going to.
Janai?
Mark is wondering
Yeah.
what the dimensions of that
Oh,
is.
all
How
right. So it is approximately 4 millimeters to 5 millimeters across. It's huge on my slide. That's why this is the lowest power of magnification I have for it. We could zoom in a little more, but I'm going to stop here, and let's see if I can just get this one last bit to see those tentacles. Um, and we are going to shift over to, let's see, we can go to bright.
are those eye spots on the hydrozoan?
Yeah, they do look like eye spots, and I don't know which one it is. I'd have to key them out. There's a lot of different hydrozoans that live in the bay, and you can key out their characteristics. But the type of organism is definitely a hydrozoan. That's the big group that it belongs to. So I'm gonna come over here and stop my share because it's time for Katie. Because one of the things that I was looking, we looked at a little bit of plankton from Lake Merritt, and we looked at a little bit of plankton from San Francisco Bay, and I think they're beautiful and fascinating, and would look at them all night long. But what Katie does is really important, and I really want her to share a bit about what she does at Lake Merritt, especially in regards to plankton and how it impacts.
Well, thanks so much, Janai. Those was amazing exploration. Really enjoyed it to see the real critters in their milieu. It was, and to get your explanations. That was really wonderful. So I'm going to talk a little bit about plankton monitoring. And before I do my slides, I wanted to share with you a picture of plankton that I took just about a week and a half ago. So you can see how a point that I would like to bring out is how different the plankton can be at different times. So let's see how this works here. Yeah.
There's a couple of bars in front of it.
And they should hopefully disappear.
Cleaning into our roofs to meet.
There we go. I think we're hearing something like the TV in the background. So here we can see some different organisms. You can see some of the little dots moving around, and those are dinoflagellates. But we see a lot of these tubular things that look like centrifuge tubes, and they're called tintinids. And if we look here, we'll see two of them coasting around together, and they are probably exchanging genetic material. I know someone had a question about sexes, and these are not male and female. It's a much more complicated system, but they do engage in mixing up their genes. And we can see some different phytoplankton here. We see a chain here, and you see some rotifers. So we can see that it can be different, just a week or two apart can be quite different. And now I want to talk about some general about plankton monitoring at Lake Merritt, which is something that I have found to be really exciting for a very long time. So I think Janai has brought up the problem of HABs and how everyone is concerned about HABs. And this is a big question that people have thinking about plankton in Lake Merritt. What happened here in 2022 to bring about this horrible fish kill? Now, I just, by way of introducing myself, since I never get to introduce myself, I'm a teacher, And I have a background in science, and I actually worked as a classroom teacher all of my career, 25 years at Oakland High School in downtown Oakland. Um, and after that, I founded a 501c3 nonprofit so that I could continue to teach. So I would like to outline a couple of questions here. What is monitoring plankton and why is it important? Who monitors Lake Merritt plankton today? Who monitored the plankton in Lake Merritt in the past, and can we learn anything? And what have we learned, and how can you get involved? So these are my questions, and I'm going to show you a bunch of slides that they're going to jump back and forth between these questions. So this one is a slide that was made by one of our California naturalists this last month, and it's about monitoring plankton in Lake Merritt very recently. And I'd like to give a shout out to Vicki Woolworth for making this slide. I added a few little things to it, but one of the one of the important things about monitoring is that it's making a regular observation of the plankton using reliable tools that can be used by other people to make similar observations, and that the information is shared with others so that we have a baseline for comparison going into the future. And Vicki did one of the last plankton collections for the Joint Water Board's monitoring program, which is focused on looking at possibly harmful species that might be in the water. In particular, cyanobacteria are involved. These tests are done all over the state, usually during the recreational time of the year, from maybe May through maybe November. And Vicki did the November one. And in the course of about 3 years that we have been doing this work at Lake Merritt, we have not found concerning levels of toxins in the water related to either the harmful algae or harmful cyanobacteria. Very little cyanobacteria, not none, but very little in Lake Merritt. And that's something that people did not know before somebody did monitoring. This gives us a good picture. Now let's see. We will now take a look at historical looks at plankton in Lake Merritt. And people have been looking at the plankton in Lake Merritt really going back into the 1970s and probably earlier. There's no way I could do a comprehensive gathering of things, but I would like to give a shout out to one of my teachers, Charles Ford at Merritt College, who taught a course in lake ecology and estuary lake ecology, and took his students out to look at the plankton. He looked at the plankton over the year and found certain things that found that the kinds of species of plankton that you see in Lake Merritt is not always in sync with what's going on in San Francisco Bay, but there are sequences that are similar. For example, in the late fall, we would see a lot of this plankton called Chaetoceros, which is a diatom with lots of spines on it, and is generally not a harmful algae, but sometimes the spines, if it gets to be really thick, can damage and irritate the gills of fishes and probably other organisms. And then later on, early in late December and January, we start to see skeletonema, which is another kind of chain diatom. So we see a pattern going on. He also saw dominance shifts between different kinds of plankton, phytoplankton, around the year. So he kind of this study and studies similar to it, there were many of them done including this one that was contributed to a Lake Merritt, a big Lake Merritt management project. They found that plankton in Lake Merritt changed a lot, and it tended to have really high concentrations of plankton compared to the estuary. Now we're going to scoot ahead to another group of people who monitored the lake in the past. These are my high school students from Oakland High. And that academy is still operating at Oakland High today and doing some water testing. During the time I was there, we did a lot of plankton viewing as well, almost on a weekly basis during the school year, which handily tended to coincide with the peak periods for plankton. And this group of students were so we didn't know the species, but we would take a picture of the plankton under the microscope and classify it as to whether it was little dots, whether it was little threads, whether there's big chunks, or where it was mixed. And the students analyzed this over the school year, fall, winter, spring, and summer, and found some patterns that were there, which actually are quite similar to the patterns that we see today in the monitoring that we do at Lake Merritt, both monitoring that is done by professionals and monitoring done by community scientists like Rotary Nature Center Friends. And here in the corner is a picture that was taken with my classroom microscope by students. It shows an euglenoid back in 2006. There are many, many images that need to be looked through to get an idea, or could give an idea, of the changeover of a plankton in Lake Merritt. Moving ahead, another interesting project that we did was with the California Department of Public Health in the Richmond lab. They have a community monitoring program that we participated in from 2022 through 2024, when we were told that our samples did not contain enough of the target harmful species to warrant the effort and the time that their lab had to put in. Their lab is focused on harmful algae that impact people, specifically people who are harvesting shellfish. And also they look for toxins that get biomagnified in the food chain. But here are two students, high school students, and they have collected some plankton in those tubes. This happened to be skeletonema. And it happened to be January, which is when we see those peaks of skeletonema. And Matt Scanlon at the Richmond lab was able to share with me his profiles of the plankton that were in each of the almost weekly samples that we had during that period of time. And we could see patterns and we could see differences. It's not all exactly the same, but we can see that there are a lot of changeovers in the type of plankton, and that it happens in some kind of an order that is understandable. At this point, I'd like to give a shout out to Dick Bailey, Dr. Richard Bailey, who also observed plankton and algae, seaweed growth in Lake Merritt, and put together a really wonderful article called A Year in the Life of Lake Merritt. I suspect that we may want to change that or adapt it as we go forward. With climate warming, things may change. But he identified the months of the fall months and the late summer and fall as the season of plankton, going into the early season of the early winter season as being the season of plankton, as opposed to the season of algae, which would be the algae that are large and leafy and whatever. We are currently having monitoring done by the city of Oakland and the Lake Tech Corporation that has the buoys that you'll see out in Lake Merritt. And they don't monitor the plankton directly, but they do look at chlorophyll. And it's just a wonderful source of information and really trying to put together many of the pieces that try to understand what is the normal fluctuation of plankton and when things start to get out of whack, as they did during the harmful algal bloom. And we're going to scoop this to here. This is another very interesting and not usual plankton bloom that we saw in March of 2024. It looked really bad. You can see the water was reddish and it was quite thick. And there was a large peak of chlorophyll, which is the photosynthesizing pigment that the phytoplankton use. And everyone was very concerned that this would have been a return of Heterosigma akashibo. But Janai and I were down there and we got a scoop of the a plankton toe and a scoop of the water, and it turned out to be an interesting organism, a cilia called Mesodinium, which also blooms in the bay sometimes. And it was here at Lake Merritt. It was not the bad actor that everyone had feared. So it's important to check out situations like that. So plankton monitoring is really important in that way. So I have not covered all of the wonderful organizations that do this. I'll have a wrap-up slide in just a minute, but I'd like to share one more. Rotary Nature Center Friends does plankton monitoring all the time in our in-person events, and you could be part of that and contribute to gathering information. You could help do the plankton tow. You could help look at and take pictures of the plankton and share them on iNaturalist. iNaturalist has projects looking at the organisms that are in all around us, and I believe there is a project related to plankton in that would cover the area of Lake Merritt. So contributing observations to iNaturalist is one way that you can personally add to the information that we have, monitoring information about plankton. So to quickly wrap up some thoughts that I have. What is monitoring and why is it important? So monitoring is planned and repeated observations using reliable tools and sharing methods and results with other people. It's really important because we need to have a baseline for comparison. When something goes really awry, it will be really helpful to know how it normally is. And it's also important because Lake Merritt is important to local people and regional people as a recreation area. And also, as Janai mentioned, it has the plankton that are the bottom of the food chain, that contribute to fish, that contribute to the bird population we all enjoy, and goes all over the continent and even beyond. And it's also an important window on the planet. As climate warming proceeds, it will be really important to have a record of how things change. So who's monitoring Lake Merritt plankton today? Many agencies of the government, nonprofits, iNaturalist, and community scientists, and some institutions of higher learning. Who monitored the, whoops, thank you to Ed Wilson there. Okay. Who monitored Lake Merritt in the past? The Merritt College, Environmental Science Academy. They're the county organizations that contributed to the Lake Merritt restoration project, engineering companies, just a wide variety of organizations back in the 1970s and 80s were involved in collecting this information that will be useful to us today. So what have we learned? Well, we know that plankton blooms are pretty frequent. They're fast changing, and they usually resolve without fish kills. There can be a fish kill without a plankton bloom, And there can be lots of plankton blooms without fish gills. And the other thing is that we have learned that there is a lot we do not know. We do not know what causes the turnover of dominant species and how they interact and why they compete, or is it just the conditions? There's lots we just don't know, and it would be wonderful and useful and fascinating to follow it up. Thank you, Ed Wilson, again. You can become involved by supporting science and conservation locally and nationally, environmental education at Lake Merritt and beyond, and come out and help monitor the plankton. And I could not resist putting in this slide by Edward O. Wilson, naturalist, visionary, conservationist, scientist, and writer, who actually died in December of 2021. But his foundation sent us at Lake Merritt congratulations and say that we have important history and we're doing great work. So 3 cheers for urban biodiversity. So thank you so much for that, Ed Wilson, and thank you to everybody who comes out and is interested in plankton. So we are 5 minutes after our official ending time. I'd like to go through a few ending slides. I want to thank everybody for coming, especially Janai, and all of the people mentioned here who are contributing to our understanding and enjoyment and conservation of Lake Merritt. I'd like to thank our supporters, the Alameda County Countywide Clean Water Program, State of the Estuary, California Naturalists, Lake Merritt Breakfast Club, New Voices Are Rising, Save the Bay, iNaturalist, City of Oakland, the Public Works, and Parks, Recreation, and Youth Development. All of these, we couldn't do anything the way we do without support. And again, we are an independent 501c3 nonprofit. Our programs, Lakeside Chats, are recorded and rebroadcast on the City of Oakland's KTOP TV, and you can go to their website and search for the rebroadcast of the week. Broadcasts are done on Sundays from 6 to 7 p.m. And the first 2 Sundays are a collection of older programs. We have now probably 65 programs, because we have a few more than just the current chats up there for that may be shown on any given week. And then the last 2 Sundays were devoted to that month's Lakeside Chat. Coming up. Yes. Okay, so in March, Charlie Onorati from Save San Francisco Bay is going to talk to us about wetland restorations around San Francisco Bay, including Lake Merritt. And so that should be a wonderful presentation. We have 2 restoration or demonstration restoration places at Lake Merritt, and it'll be really helpful and interesting information for us. Plankton monitoring is taking place now on Thursdays from around 9:30 to 10:30 at the Lake Merritt Boating Center dock. If you want to just check it out, just drop on by. I'm going to put up an Eventbrite so that you can sign up, and then I can get back to you to just confirm that we are in fact meeting. But I want to make sure that we regularly monitor the plankton. There will be another Mornings in the Marsh program. We'll get to schedule that one. We have 3 really great programs scheduled here for coming months. We are going to hear from Lisa Owens-Vianney about how raptors are the solution, not chemicals, in controlling pests like rats, mice, etc. And we'll be hearing from Christina Fall, the Goldman Chair of Environmental Science, Mills College at Northeastern University. She's going to talk to us about urban estuaries and her research. And then in September Andrew Alden will be coming back to talk to us about the lakes of Oakland, so not to be missed. We'll be sending out information to you. If you enjoy what we do, please consider making a donation to Rotary Nature Center Friends. All of our funds go to providing for program and for the Lakeside Chat programs. And now we are providing support for the California Naturalist Program and making it available to everyone in our community. So we would appreciate anything that you can donate in that regard. So Rotary Nature Center Friends is an all-volunteer nonprofit in the center of Oakland, dedicated to interpretive education and science for all people of Oakland. So at that point, we have gone over, but Janai is still here, and it's a great time to ask her some questions and just to engage in general discussion. So please raise your hand if you'd like to say something. You may unmute and we'll call on you. And you can put your question or thought into the chat as well. Okay. Excellent.
All right. I'm here and I have there's just so much to see in the plankton. It's really hard to stop looking. I found the arrow worm I was looking for before, and yeah. Anyway, there's a lot to see. If anyone wants to look at more plankton or has any questions, I believe I adjusted it so you can unmute yourselves if you like. And I am going to share my secondary camera again. Let's see if I can do it without... Oh, before I do that, I'm going to do this. My closing slide. So there's 2 events coming up that I'll be at with the Microscopical Society. And one is Pond Scum Night in Oakland at Merritt College. It's going to be on Tuesday, February 10th from 5:30 to 7:30 at the college in the microscopy department. It's a very casual hangout where we look at whatever people are curious about on the microscopes they have. So if you have something you're curious about and you want to bring it and look at it on a microscope, it's happening in Oakland. And then there's Microscopes and Chill at a tavern in San Francisco the following Tuesday from 5:30 to 8:30. So that's my last little bit there. Oh, I see a hand. Hey, Adrian, unmute yourself. Go for it.
Hey there, Janai. Thank you for the presentation. Thanks, Katie, as well. I feel a little silly because I always have thought of plankton as like one group of things, but this from your presentation, it sounds like phytoplankton or plankton is more of a lifestyle than it is like a category of life.
Yeah.
Is that an accurate?
That is an accurate statement. So lifestyle can sometimes imply choice. However, plankton don't choose. They are the way they are. So yes, I guess lifestyle works, but yeah.
Lifestyle decided by evolution.
Yeah, yeah. The niche they fill.
Cool,
yeah.
Thank you.
Uh-huh.
Oh, Jim Covell had a really interesting question. And also, let's see, Mark has a question about the kind of microscope you use. I imagine microscopes is...
Microscopes is?
Yeah, there's so many.
I do.
There.
So what I was using tonight was an Olympics. Is a BX51. It's a lab quality microscope with a View 4K camera attached, going through a CamLink adapter into OBS to share on Zoom. It was kind of like a hodgepodge of ways to get there, but I have a field scope I can take with me when I'm out at the lake and I want to look at stuff right away. I have battery operated microscopes that I can bring with if people want to share, because you don't have to hold them like binoculars. You can just set it on a table and look. So there's a lot of different types of microscopes that, depending on the situation.
Yeah, so at Lake Merritt, when we have our events, we use a really simple microscope with an adapter that you can put your iPhone or your smartphone into. If it fits into the little holder, you can come away with your own photos of plankton from Lake Merritt. So that's another choice that you can do.
So let's see.
Janai had a question about what the vertical migration of plankton might look like in shallow bodies of water, like Lake Merritt, and San Francisco Bay.
So I don't have vertical profiles of plankton taken over 24-hour periods in a specific place where you can do this type of analysis. It sounds like a really great question. The thoughts that I have about it, which are not scientific thoughts, just based on what I've looked at over time, the thing that seems to affect the plankton that I look at near shore the most is the flow of water. If the tide is coming in hard or going out hard, or it's windy and there's a lot of mixing, I'll see things differently than if it's a really slack day, time of day. There's not a lot of movement in or out. So because it's near shore or it's inland, the places where I'm looking at the plankton, that seems to be have a greater impact than the time of day. But that said, there aren't a lot of places I go to after dark and collect plankton. It would be a fun thing to do if there's a group of folks that are interested. But I don't think I would feel comfortable going on my own to the water's edge in the middle of the night, quietly dragging a net back and forth to see what's there. So we'll make it a big group event, and then I'll feel comfortable doing it. Do I have any slides, pictures of bioluminescent plankton to show? So the sea sparkle is a dinoflagellate, and we saw it. We have a slide of it. The light that they give off is very dim, and you need either special cameras and or equipment to be able to see it on a microscope. Yes, Edith. So if you think about it, the dinoflagellates, when they're not in these huge groups and these giant blooms that make the surf sparkle along the coast, if there's just individual ones, each individual one is giving off the tiniest fraction of light. And I know with Noctiluca, it's a luciferin-luciferase reaction. It's a chemical light giving off reaction, and it'll do it, and then it'll like use up its store and be done. But when there are blooms of them, there are so many, they don't all use up all their glowy bits at once. But because it's such a tiny increment of light, you would need a special camera. And on the microscope, mostly what I'm doing is I'm shining a bright spotlight on things, trying to be able to see the details in the organism. And so the light that they could possibly give off would be many, many, many times less and more dim than the light that I shine on them. So I know people have done it, but I don't have that equipment. Best thing I can recommend locally is sometimes the shores of Lake Merritt on a moonless, foggy night at the right time of year when the right organism is blooming, you might be able to see some bioluminescence. Otherwise, another place would be Tamales Bay is another place that has some beautiful bioluminescence, and they are far enough away from the lights of the city. It's very dark, especially when it's moonless, and you're more likely to see them there. I was
Yeah. I'm going to try to find this. There's on iNaturalist, there is a map or two showing where in Lake Merritt the bioluminescent areas are, based on observations by Damon Ty going out in kayaks in Lake Merritt at night. And I think he has maps from several different years. I have to try to find it on iNaturalist. So I might have to put that into a post chat letter because it'll take me probably a long time.
I'm trying to switch. There we go. So this is now live on my microscope again. This was San Francisco Bay plankton. While you're doing that, Katie,
Okay, I'll do
there's
it.
just so much to see. It's really hard to stop looking. There's a lot. There are different shaped diatoms and dinoflagellates near the Golden Gate Bridge, and the tide was moving much. It was flowing very strongly when I was collecting the plankton near the bridge by the warming hut. So it was, I had a different sample, not just from a different location, which invariably has a very different profile, but a lot of the organisms are in the Merritt plankton, just not in the same abundance and all the what's the word, in the same ratios. So we have more dinoflagellates here. All these ones with the golden brown are diatoms. This little ring here is a spiral of diatoms. These are diatoms here in a chain. Yeah, these here are diatoms. I think that's skeletonema there, which is one that we had blooming at Lake Merritt not too long ago. Let's see. There's just so much. The problem is when I switch over to this camera, I can't see the chat. So since the official talk is over, if you have a question, unmute yourself.
So I found the link.
Great.
Yeah, this is to actually an issue of the Lake Merritt Institute's newsletter, The Tidings, which I edited at that time, and has a big article and with some photos of the bioluminescence areas in Lake Merritt discovered by Damon as he went out in his kayak. So if you click on that link, you'll see that issue.
Nice. So someone had also mentioned reproduction and diatoms as they divide, because they're single-celled algae. As they divide, I'm going to switch back to my other screen so I can show you with my little arrow. As they divide in two, because they're single cells, this is ditilum, it's a diatom. This is also ditilum, the same kind of diatom. But when they divide, every time they divide, they get a little bit narrower. And so this one is huge, and it's a big baby. And then the skinnier they get, the older they are, the further along they are in that division series, dividing and dividing and dividing. So, so it's almost a reverse of what we expect. We expect adults to be big and babies to be small, but in diatoms, the youngest in the series, in the reproductive series, are huge, and then the older they get, the skinnier, the thinner, the narrower they get. So that's kind of a fun one to see in action right there. I was going to see if I could see any dividing, because sometimes you get lucky and you get to see. Oh, this actually has one that's very old, or very far along in its series of division. So this one here is also a ditilum. So this one and that really big chunky one that was so beautiful, that little baby over, I lost it. Where is it? It's here somewhere. There it is.
Oh yeah.
It was huge in diameter compared to the other one. So this one here, you can see how big, how wide it is. And then.
So Karina had a question about magnification. What magnification are you using?
Magnification questions. Can you unmute yourself and ask, or do you want me to click over and read? What magnification?
I can try. Hi, everybody.
Hi.
I'm part of Shark Stewards, and we do observe plankton at Aquatic Park Lagoon in Berkeley. And I'm a former biologist. I used to work at UCSF. Now I'm teaching science for homeschoolers, and my daughter is an intern for Shark Stewards. So I do have about 12 microscopes in the house. I used to do a lot of microscopy. I'm very rusty on identification, but I can manage the basics. So that's why I was very interested to attend this meeting, because I want to find someone who actually knows them, because I am not, I really don't know details, basically. So I ask about magnification because your microscope is very different from what I use. It's a fancy one, and I believe it's the minimum, right? It's probably 4 and whatever you use for objectives.
Yeah. Yeah. So
they're huge. Diatoms are huge.
Diatoms. So marine diatoms can be huge. The ones in the ocean can often be huge. The freshwater diatoms are often much, much smaller. And there are marine diatoms that are very tiny, and they are often overlooked because they're so small. So there's like really tiny Thalassiocyra and other are really very small diatoms. But so magnification on a screen like here is tricky because it goes the image. If you were looking through the microscope, you take the objective, the part that points down at the stage, and that the number on that is like 4, 10, 20, 40, 100. It's one of those numbers usually. You multiply that by the number on the eyepiece or the ocular, which is usually 10, not always. So like 4 times 10 is the lowest power on this microscope. So that would be 40 times if you were looking through the eyepiece. But then what happens is it goes through a photo tube that has some magnification in it instead of an ocular. And then it goes onto a camera sensor that is a certain size that then is run through a video capture card and onto a screen, and I have no idea what size screen anybody else is using. So it gets really complicated really quickly when you're using screens and cameras and microscopes. But if you're using your eyes in person with a microscope, that's like the easiest way to do it. If not, scale is usually more helpful. So things like right now, I'm using my 10 power objective. So if I were looking through my microscope, it would be 100 times actual size that I would be seeing through the microscope. It would be magnified that much. But I can tell you that on the stage, the width of my screen is about 2.5 to 3 millimeters ish across. So it's the scale that can be more useful when you're trying to share with images, and that's why a lot of scientific texts have scale bars, because you never know how big or how small that image is going to end up. But if there's a scale bar, that can help you figure out the size of the actual size. I don't know. Does that help?
Yeah. I was just very surprised that diatoms, usually I see them smaller, but here they're gigantic. That's why I was wondering.
Yeah. It's because cameras and screens and capture cards, and it gets, yeah.
Thank you very much.
Sure.
Well, by the way, if you're planning the night foray, we're very on board. we just had a Sonoma Mushroom Ecological Association camp, and Damon was one of the guys who was leading the foray, and he introduced me to his flashlight, which is amazing. So I've got 365 nanometers, and now we would like to go to aquatic during the night, and we're trying to get a group, so we're not alone. So we have maybe like 3-4 people, but we're trying to figure it out when, because everybody are busy.
Yeah. Tide pooling can be really interesting with black lights is really the thing to do. If you have a safe place to go to tide pool after dark, that you know well enough that you're not going to get caught if the tide comes in, because it adds a layer of risk being in that environment after dark. So that can be a really lovely thing to do. With microscopic organisms, it's if you're interested in seeing the fluorescence, which I think is what you're referring to with Damon's flashlight.
Honestly, he was just showing chlorophyll A absorption, which I never saw with normal $10 UV flashlights. His is a bit shorter wavelength. So usually 390, that's what the least expensive one would be. His was 365 and exciting chlorophyll A, which was amazing.
Yeah, the 365 nanometer flashlight. For people who don't know, if you're looking at things using different wavelengths of light, fluorescence is when they'll receive and absorb a certain wavelength of light and then re-emit a wavelength of light of lower energy and a longer wavelength. And it's a really amazing process. At Merritt College, they have an array of fluorescent microscopes. So if you're interested in, you're welcome, Jerry. If you're interested in looking at microscopic organisms with fluorescence, I highly recommend checking out the offerings at Merritt College, because they have the tools to do it safely, and it's a community college, so you kind of can't go wrong. So lots of fun stuff there. We'll be there on Tuesday doing pond scum night. So, which, even though it's called pond scum, you can look at anything. But pond scum is usually really cool. I like the algae. Katie was mentioning Cattosaurus blooming, and we have Cattosaurus here. Let's see if I can. There's some Cattosaurus with those little tiny. Let's see. I'm gonna switch to my other screen so I can point them out.
Janai, are we back? This is San Francisco again?
When? This is San Francisco. Sorry.
I thought so. Yeah.
Yeah, I was just looking for something different and interesting. And these are the Cattosaurus. They've got these like long little setae or hairs that come off of them. They're made of silica, including these little extensions coming off of them are also silica. And they're really quite amazing, but they can be a mechanical irritant to some fish when they're when there's a lot of them in the water, because those little tiny cell extensions can get stuck in the gills of fish. So, skeletonema. Did you want to try and look at that dinoflagellate that we have at Lake Merritt right now?
I would very much like to see that. Yes.
Would you? I'm going to see if I can find it. I know I have it on a slide here. I just can't remember which one I stuck it on. So bear with me for a moment while I play. Any other questions?
Yep. Sorry, I was trying to look at Cattosaurus. Could you, please?
I can't. Perhaps Katie can.
Well, I got it wrong when I first posted it on iNaturalist, but I think it is Chaetoceros. And then there are many different species of it.
Okay.
Let me try to put it. I'll put it in the chat. The way I understand it, I think it's right.
You're correct. You're correct. Thanks so much, Katie.
Sure.
All right. Let's see if I can get this back and focus. It'll take me a few minutes here just to like ramp up and make sure I've got the right ones. And why do I have that's So that's phase. This is bright field. There we go. There. It'll take me a minute. Any other questions while I fiddle with the microscope?
Actually, yes. When is this event at the college on Tuesdays?
So it's not every Tuesday, but it's going to be, here, I can see, maybe it'll let me. Will it let me? Portion of screen, share. There we go. Here's the information about it, and it's the Microscopical Society and Merritt College working together to bring you fun. But there we go. I'm gonna.
Thank you. Are they showing their own material, or can we bring our own recordings?
If you bring stuff, you are responsible to take it with you, because we
I
can't.
have already like pre-recorded videos and photos.
Oh, videos and photos. We're gonna have live material and a microscope connected to a screen. So I don't see why you couldn't bring your photos. I just don't know what the facility is for sharing them with the group other than just individually. I haven't encountered that yet. I guess we'll find out. Sound
So
good?
if we bring in our own sample, we will be able to look under the scope and possibly get help to identify.
Help to identify is always hit and miss, because you never know who's going to be there and you never know what you're going to have. So, we can do our best to help and support. But a lot of times we will point you in the direction you might want to go for more identification, and not necessarily be able to do it for you. There are people whose entire careers of study have been around identifying things. So it's we like to support people in their learning, and like where to find the information and I picked the wrong slide. How to get like the next step, right? These are the wrong ones, but I'm gonna start with that and then I can go to the other slide. Did I lose my screen share when I switched away? I did, didn't I?
Yeah, you did.
Okay. Here we go. Then I will switch it back. Any other? Let's see, where am I? Share. Second camera. Let's see.
I have a question maybe to you and maybe to Katie as well. If, let's say, we're trying to keep track of plankton in lagoon, inner lagoon, what would be the best way to do it? Again, we don't have that many people involved and not so much time. I mean, we can probably take samples like once a month, and I can just sit with microscope and make photos, but maybe there is a better way to do it.
Katie, do you want to try?
Oh my goodness, I was just typing a message to Karina and saying, I would love to connect around sharing plankton monitoring projects and photos with you. One of the things that I noticed in one of those older documents was the assertion that the plankton in Lake Merritt was more like the plankton in Aquatic Park than in the the Outer Bay. It would be really interesting because I know plankton is being collected at the Shorebird Nature Center from time to time. It might be kind of interesting to see what we're getting at the same time. I would love to see what you're doing and share photos and so forth. It's for us, it's just when we can get out there. But I've decided I'm going to, during the season of plankton at least, I'm going to be going out every Thursday, and I like to post it on Facebook because it's easy for me. So I've been doing that. If anybody's interested in following that Lake Merritt plankton amateur photography results, I'll put our Facebook thing in here so that you could check it out.
Also, I posted my email if anything comes up, like night forays or any other trips. Please let me know.
Thank
It's
you.
a little busy right now because I have close to 50 students and they come to my house, so I teach them in groups of 8. But I can escape if it's like Tuesday evening or weekend. I can try to escape. At least I will make my best to escape and meet.
Wow, that's a wow.
So this is just to like, while I'm trying to track something down, this is plankton from Lake Merritt, and it's got a lot of these little rotifers. The salinity must be a little lower because these always seem to me to be around when the salinity is a little less. But
it's about 21 parts per thousand.
Okay, so like under 30.
Oh yeah, it will be under 30 after the first big rains for a while. But if we have a really wet spring, it goes down to like 10 and can sit there for weeks. And oh wow, look at that nice worm. Woohoo.
It always happens when I'm not trying to find them. As soon as I stop trying to find them, they all show up for whatever reason.
Really? Look at all those guys. I didn't find so many worms in my sample, but
there's one of the things that I do is I will often look for the plankton in the jar. I'll sample. Where am I? I'll have a light and I'll shine a light at the jar, and plankton are often either attracted to or repelled by the light, and I can select more. I can be more selective in my selection.
Yeah.
So I was going to try and find those. There's the dinoflagellates, but there's just so many worms. It's a little bit harder. Okay, there's copepods. There's so many little rotifers. Oh my goodness. There's a great big copepod. Look at that.
Yeah. Well, these are great.
Now they show up afterwards.
I never use chemicals to slow them down, but I read that you can add something so they can kind of stop doing that. Usually I just make the slide a little bit drier if I need to show to kids.
I used to use
what
Gatorade.
Can you add to slow them?
Back in the day, we used to use a little bit of Gatorade. I'm sure it affected them a lot, but it did slow them down enough so that you could, of course, it would obviously eventually distort them.
Kids would love to try that.
I mean, it allowed us to see them. This was the high school class. But man, I got a microscope in the early 2000s. It was a digital microscope, and we took pictures every week. And I have years of slides to go through. So now that I'm kind of focused more on looking for seasonal patterns or seeing, you could actually take a look at all the Januaries and then take a look at all the Februaries and just make a list of what you're seeing. Not truly scientific, but kind of, it would be fun to look at.
There's a little clump. Ecdysis. It's going to pop out of its. I'm getting bumped around so much. Those rotifers are really photobombing. Any other questions while we're playing around here at the end of the night? Looking at plankton.
Kathy, I put my email in the chat, so let's make it happen. Let's meet somehow. We're having our next beach cleanup on February 15th, which is kind of our time to gather if, someone would like to come.
Great.
We did cleanup 3 weeks ago, and it was the next after king tides, and it was horrible, and we decided to do one rather soon, so February 15th, our next one. A lot of garbage at Aquatic.
Yeah.
I don't know if Lake Merritt is affected as bad as. Do you guys get a lot of plastic, a lot of any other stuff?
There are barriers for that keep a lot of the trash from making it to the lake when they come down the tributaries. Katie, you can speak to that better than I can. The Petra barriers?
Kitty, I think you muted.
Uh-oh.
Yeah, I was going to say the Lake Merritt Institute goes out at least twice a week with nets and barrels and pulls out trash. And then there's another organization, the Trash Falcons, that's very active, pulling out, specifically focused on pulling out trash. And there's a lot of it. There really is a lot of it. Sometimes there's not. And then there's a trash collector at the tide gates. There's little trash barriers at the entrance of the lake from major storm drains. And then people go out and fish out of those areas and are able to remove a lot. But some of it sinks and, eventually it's going to go out into the bay.
Well, at least it seems like there are a lot of different organizations who are around the lake. In
Yeah.
Aquatic, I don't think we have maybe 1 or 2 groups who actually does cleanups. Shark Stewards, us, we do it once a month.
Mm-hmm.
And I think another organization, Waterworks, they do sometime,
Mm-hmm.
but they're on the other side of the lagoon.
Uh-huh.
Now there's all the plankton, right? No matter what I do, I prep slides and
Whoa.
it always happens.
They come out late. They get
it's like you're going to talk for 20 minutes and you're going to make sure you get plankton off the screen. I'm like, try. And it's only later that the plankton settles and I can select and pull things. It's so frustrating.
These are beautiful.
Yeah, there's a lot of lovely stuff today.
Mm.
Oh, it's beautiful.
See who's in the background there. Yeah, the microscope I use has a DIC optics, and so that's why I'm getting this kind of secondary coloration. I can adjust it so we can see some more of it's been eating dinoflagellates though.
You can see
Look at that.
Yeah,
there's the dinoflagellates in the gut. All lined up in its gut. Oh, there we go.
Story.
Yeah, but I think so. When some of the dinoflagellates get stressed, they pop out of their theca, and so sometimes I don't know if it's the dinoflagellate was stressed and so it popped out. Which is why I was looking for it. Oh, look at the little fine feathering on the tail of that copepod.
I'm so jealous. Your microscope is amazing.
Well, it's a lot of fun. There's so many ways to adjust. It often just takes more time, and there's the dinoflagellate somebody popped out of.
It's the wall or the theca.
The theca, yeah.
There's a
one.
cutie. There's one. Oh, yeah.
There they are.
Whoa.
They're going to swim around too fast.
Yeah.
Yeah, they're such great environmental indicators too, because they reproduce so quickly. The plankton, when it changes and then falls down into the sediment, because they respond to changes in tide, temperature, sunlight, salinity, freshwater input, saltwater input. I mean, like all these things impact these populations.
Yeah. What's that one? Let's see.
Yeah.
There was that one that somebody, was it gymnodidium or something?
So there's lingulodinium is one of the little ones. Gyrodinium is usually bigger. Gyrodinium is quite large as far as dinoflagellates go.
Okay.
I mean, quite large is a relative term. They're all tiny, right? But
right.
I'd have to check what that's a neat little rotifer. So yeah, it's not gyrodinium. It might be a lingulodinium because it's got that strong.
That's the polyhedra one?
Maybe. I mean, there are so many different, right? Like getting to genus is one thing, maybe. But getting to species, it's just, unless they've got really obvious features, I would want to see it and go closer with a microscope, because so it looks like the cingulum, which goes around the waist, is nice and even. It's straight across, which helps when you're identifying, right? It's not offset, and it has one apical point, and then it looks like it's pointy on the other side too. So I don't know.
Yeah, it does look pointy. It looks kind of like a flatter point, kind of.
Well, I don't know how much longer you want to hang out in the plankton here with me, but it's almost 9 o'clock.
Yeah, I think we should probably.
Well,
oh,
thank
you're
you very
muted
much.
again.
I got somebody muted me. I was going to put in. We should have a link to the Cadella Lab phytoplankton gallery, right?
Yeah, they're great. They're a great
I always
resource.
go there. I mean, there's a lot of sophistication that that helps to.
Yeah, and even the Cadella Lab key, is really generic. I mean, it's the thing most people don't realize is to key out the organisms to genus or to species can be, you have to be able to differentiate certain features that you can't always see unless you're looking at it under high magnification with an oil objective, right? Like I can tell it's a rotifer, right? If I knew my rotifers better, I could probably tell which rotifer it was or get close because I know the ones that are local. But if you wanted to be sure you got to the right species, you would want to look at very precise features. Like on dinoflagellates, a lot of times you want to look at if they're thecate, if they have the hard plates, you want to look at the shape and orientation and ornamentation of the plates. You want to look at the way the flagellar pore, like the where the flagella comes out, that kind of shape around there is important for identifying dinoflagellates. So whenever somebody goes to species, I'm like, ooh, what did you see? What was the key? What characteristic
Oh, wow, look at that guy.
You know, did you find that allows you to make that
Uh-huh.
determination? Because I'm always curious. I don't know enough of those. Some things I do, like Dityllum, that one diatom with those long pointy ends, it's really unique, right? And so that one stands out. Catostris is a huge group.
Right.
But they have a structure that is identifiable because of those.
Oh,
Whoop, there it goes.
that was quite amazing.
So anyway, I can
Yeah,
ramble on and on and
tonight,
on.
Thanks
this
for
is
the
amazing.
yeah.
So
Thanks for the
basically, if
amazing
we're
imagery.
doing any kind of report to city or to whatever organization for grant, maybe it's okay just to provide common name like dinoflagellate would be sufficient, or do we go to genus?
Every monitoring program has their own protocol. How the sample's collected, the kind of net that you use. Is it whole water? Did you filter it? Is it preserved sample? Did you let it settle? What volume of what? Like, there's a lot of different things that if you're doing monitoring, right? And you could just do it really straightforward, like Katie was saying, you know, where like, you know, are they long, skinny things or round, short things? Are they big, blobby, moving? You know, like have it be, you know, really.
Yeah, I was impressed. I think now we know a little bit more, possibly what was involved in those things that we used in the high school class, you know, like the moving dots and all that. But it was useful to, you know, to classify what we were seeing in a way that everybody could use easily, quickly. But I think, you know, if you could probably, you know, send something off to a lab or maybe make some photos and send, maybe Janai could help out, or you can put it on iNaturalist because they have people there who are quite knowledgeable and they will get back to you, and they're very kind and willing to talk to you about your picture. If you put something up on iNaturalist.
Yeah, somebody asked about moving and vibrating and emitting sounds. So all sound is vibration, and the sounds in the water are more important than we realize. They don't have ears the way we think of ears, but they all respond to vibrations in their environment. And I have not read any current studies about plankton and sound, but I do know that it is something that people are studying because the sounds in the ocean are changing. And as the temperature of the water changes, how sound propagates through different temperatures of water changes. So it's a really interesting area that people are looking into. So
Really great question. Yeah.
Yeah.
Well, Janai, this has just been amazing. We could keep you here for another hour or more, but I really appreciate your sharing all of, you know, all the work, the prep, going out and getting the samples in 2 different places, and yeah, it's really wonderful. I really appreciate that, and I know for me, you know, it makes me want to study more about plankton and maybe get a little bit better at my identifications. And also makes me feel like, you know, yeah, Lake Merritt is a very special place within the region, and it's so accessible. We're looking to have, I think soon we'll be able to have a slight indoor place to go to look at our plankton. That'll be really nice. And yeah, it's really great to share that with everybody, and it's something you do so beautifully. So I really thank you so much for tonight. It's been wonderful.
Thank
So
you.